Hi, so it is my pleasure to welcome Jeanne Stachowiak at our today's kitchen table talk. I actually first met Jeanne about 2 years ago at the GRC IDP meeting, where she presented a fascinating story where she was assigning IDRs roles in sensing the membrane curvature. Jeanne is actually a mechanical engineer by training, and she has earned her degrees at the University of Texas in Austin, and then her master's and PhD at UC Berkeley. Currently, she is an associate professor at her alma mater back at University of Texas in Austin. She's in the Department of Biomedical Engineering. Jeanne's new work, which I think she's going to talk about today, shows now new involvement of membraneless organelles in the regulation of membrane biogenesis. So I think that's pretty cool. So without further ado, thank you, Jeanne, for accepting our invitation. And we look forward to hearing your story.
Great, thank you. I'm really glad to get to talk to you all and meet you. Great, so I'm going to share my screen. All right, can everybody see? You see the slide all right?
Yes.
Yeah, okay, great. All right, so it's really a pleasure to speak with you guys today, and I'm really excited to hear your thoughts on the work we've been doing. So for the last several years, we've been thinking about the roles of disordered proteins in membrane traffic, in the assembly of trafficking vesicles at the plasma membrane of all types of cells. So this is a field that for decades really has been very much dominated by structure-function relationships. There's a lot of structural biologists, and to some extent, the history of structural biology is really intertwined with the history of membrane traffic, with proteins like the bar domains and clathrin and many other structures being solved out of this field. So it's really a perhaps strange, unusual perspective to think about disordered proteins in this pathway, but as you'll see, they play some important roles. Okay, so I'm going to start. Oh, let's see. Oh, now I have to figure out how to get it. I think, how do I get it to advance slides when I'm sharing screen? Oh, aha, okay, figured it out. So I'll start with a little bit of background on this field, and then I'll tell you about some of our earlier work, thinking about how disordered proteins as individuals function in this field, or function in this pathway. And then I'll move into how do disordered proteins, when they interact with each other, when they form networks, function. And that's where we'll come to protein droplets, which that'll be great to hear what you guys think about that. So first, membrane traffic is one of, and how membranes become curved by protein assembly is a very fundamental question in cell biology and biophysics. And many labs have made contributions in this field for several decades now. And you can see here an example of one of the most studied structures, which is a clathrin-coated vesicle. Clathrin-coated vesicles are studied so broadly for several reasons. They form at the plasma membrane, so they're really accessible to a lot of experiments. Also, the clathrin coat, this density that you see here around these vesicles, is very accessible and obvious in many different types of imaging, from EM to fluorescence microscopy. And then many receptors are specifically internalized by the clathrin pathway. So that sort of makes the clathrin pathway relevant to many different signaling processes and disease processes. But this question of how do membranes take on these highly curved shapes, maybe 50 to 100 nanometers in diameter, has really been the source of a lot of debate and work for a long time. And traditionally, there are two main classes of answers to that question. The first is there's a broad set of proteins that are known to have specific structures that can scaffold the membrane and really impose their curvature on the membrane through a curved binding interface. And here you see this structure, which is the structure of the bar domain. It's a dimer. But then we can also think more broadly about structures like the clathrin coat itself, or the cylindrical coats like dynamin that cut off the vesicular neck. So many different scaffolds. And then the other major way of answering this question has been to think about things that increase the spontaneous curvature of the membrane. So one of the primary ways of doing that is to insert something into one leaflet of the membrane that's not being inserted into the other leaflet. And here the example is an amphipathic helix is being inserted into the membrane, and quite a few endocytic proteins attach to the membrane with these amphipathic helices. So clearly both of these mechanisms rely on specific structural motifs, whether it's a scaffold with a certain shape or this inserting helix. But a really important thing to understand about many of these endocytic proteins is that they also have substantial regions of disorder. So here we're looking at the same little helix of this protein called epsin inserting into the membrane. And on the previous slide, I was just showing you the structured portion of epsin called the ENTH domain. But you can see there's also this large intrinsically disordered domain. And biochemically, we know a lot about this domain. You can see some of the binding interactions mapped along it. But physically, relatively little is known about it. And then, so that in the first bit of the talk, I'm going to talk about these disordered regions and what they can accomplish sort of individually in terms of driving the membrane to curve. But then the other thing that these disordered regions are responsible for is forming these multivalent networks among proteins. And this is an area that's just beginning to be explored. Many of the experiments in the field have really just looked at one protein at a time, but relatively little is known about these networks. So in the second bit, we'll talk about what we're learning about the networks. Okay, so first let's talk about these disordered domains. So I've already pointed out that proteins like epsin contain these disordered domains, but then many of the other endocytic proteins, for example, here's one of those that contains a bar domain scaffold, but you can see that each monomer of that dimeric bar domain also has a substantial disordered domain. And this is a protein called amphiphysin. Both of these proteins, epsin and amphiphysin, are what we refer to as adapters. So what adapters do is they bind the membrane. Some of them interact with transmembrane cargo, but they also interact with the clathrin coat. So the clathrin coat doesn't bind the membrane directly, it's recruited by these adapters. And many of these adapters have large intrinsically disordered regions. Okay, so this first little story is about how those disordered regions modify the functions of adapters. And we'll start by talking about this bar domain containing protein amphiphysin. So first I'll point out, if you're not familiar with bar domains, that they're a large superfamily. They're these sort of banana-shaped dimers, and they have a range of different curvatures. So the most highly convex bar domain are these, what are called N-bars, and they're involved in shaping endocytic vesicles and shaping the neck of the vesicle in particular. And then there's these slightly less curved F-bar domains, and F-bars are involved in the early initiation of coated pits, and they're also involved in other pathways throughout the cell, like driving cytokinesis. And then there's actually these I-bars, which I stands for inverse, so they have the opposite curvature. And as you might expect, they're involved in forming structures of opposite topology, like filopodia or dendritic spines, so things that poke out of the cell rather than things that poke into the cell. So the bar domains are really a structural biologist's dream. They not only do they have a nice looking structure as a dimer, but if you let them assemble on a surface, so here you're looking at bar domains assembled on lipid tubules, they make this beautiful helical oligomer. And so cryo-TEM can really highlight that oligomer, and a lot of these structures have been mapped. So from these cylindrical structures, the main interpretation of the function of bar domains is that they stabilize cylindrical architectures. So they've been thought to stabilize these necks. So imagine this is the endocytic vesicle down here, and here's the plasma membrane. So they're sort of responsible, it was thought, to stabilize the neck of the endocytic bud and then recruit the proteins like dynamin that are gonna cut the neck off and free the vesicle. Okay, so that had been the paradigm for understanding these proteins. But almost all the work leading up to that idea and all those structures that I was showing you were all solved without the disordered portions. And that's for a variety of reasons. It's tough to purify the full-length proteins. They tend to inhibit crystallization. So they really get in the way of structural biology. But we set out to ask, what are these disordered domains doing? Do they in some way modify the function of this amphiphysin protein? So this was the work of a graduate student, Wilton Sneed, who actually recently moved on to Amy Gladfelter's group, if you know her. So Wilton he started looking at this, and at first he just did some very basic stained EM. So this is what vesicles look like in a negative stained EM. They look like these crushed cans on a grid. And then if you add the bar domain only of amphiphysin, before you put them on the grid, you'll see these long straight-sided tubules. You can almost make out a little bit of the helical oligomer on them. And lots of people have seen that in EM. But then if you use the full-length protein, instead of seeing these tubules, you almost never see a tubule. Instead, you see all these little vesicles that have a diameter sort of similar to the tubules, but no tubules. So that was really surprising to us. Somehow this protein is breaking up the membrane. So we're trying to get more insight into that. And one of the first things we did was just to run the experiment another way in a higher throughput format. So rather than using EM where you can count relatively few objects and the interpretation is a little subjective, we tethered the same vesicles on a cover slip and used some calibrated brightness techniques to estimate their diameter distribution. So here you can see the vesicles by themselves, but then if you add the bar domain, you don't get much of a shift in diameter. They still look like dots because the resolution's low, right? We have the optical diffraction limit. So even some of those short tubes are still diffraction limited in length. But we don't get this shift in the overall brightness of the objects. But then when we add the full-length protein, we see this dramatic shift in these small objects. So somehow, the full-length protein, which contains the disorder domains, is driving this transition from tubules to vesicles. And this is really perplexing, but it makes a little bit more sense if you think about it in the context of this oligomer. So here's the oligomer that was solved in a recent paper, beautiful structure. And think about where are the disorder domains. So there's two of them on each dimer. So I've kind of put them into this cartoon down sort of one stripe along the helical oligomer. And then we measured their hydrodynamic radii using fluorescence correlation spectroscopy. So you can see about how much space one of them would take up. And then if you draw them all down this stripe, you see the problem. They really don't fit. There's this substantial overlap. In fact, you can calculate that at the surface of the oligomer, you need sort of 2 to 3 times more volume than you have to fit them all in. So what that would mean is that if they're all going to fit, they'll have to get straightened out. Sorry, did someone say something? Oh, okay. Feel free to interrupt if you have a question. So, and straightening them out, as you all are probably aware, if you think about polymers, incurs a pretty substantial entropic cost. In fact, by our estimates, that energetic cost will be greater than the cost of breaking the membrane into pieces, which is actually relatively inexpensive energetically. So our hypothesis became that this steric pressure among these tightly concentrated disorder domains is what was driving the vesiculation. But that idea has several obvious implications. Basically, by hypothesizing that, we're saying that sorry, give me 1 second. I'm going to turn off my email, which is giving us those annoying dinging sounds. Okay, good. Sorry about that. So that hypothesis has a couple of obvious implications. One of them is that we're just acting there as if the disorder domain is just a certain size, just a steric bulk with no particular specificity. And if that's true, we ought to be able to chop off the existing disorder domain and replace it with another one of similar hydrodynamic radius and see the same thing. So we did that. We made this chimera where we replaced the disorder domain of amphiphysin with another disorder domain from another endocytic protein, epsin. And we compared the wild type protein to the chimera. And here you see our shift in vesicle size distribution as a function of concentration. And if you plot these data together, you'll see they match very well. But then even a further implication is that it shouldn't matter how we attach the protein to the membrane. We should be able to just take the disorder domain by itself. And if it reaches equivalent density on the membrane surface, it should drive the membrane to break up as well. So we ran that experiment, and that turns out to be true. But to get up to the same coverage of the membrane surface, we had to go to dramatically higher protein concentrations. Before, we were talking about maybe 250 nanomolar. And to drive, sorry, gotta go back one, to drive fission of the membrane with this monomeric version that's attaching to the membrane with the histidine tag, we needed more than 10 times that much protein. So that really illustrates that the bar domain, which can oligomerize on the surface and is a dimer, is really potent at concentrating these disordered domains. And that, more than directly scaffolding the membrane, may be one of the key functions of the bar domain, which is to concentrate proteins locally. Okay, so we wanted to look at some of these same ideas in cells. So here you're seeing retinal pigmented epithelial cells, which are popular in this field because they have really big lamellipodia, so they're really spread out and thin, and you can do great turf imaging of the plasma membrane. So here we overexpress the M-bar domain, which is an old experiment that a lot of people had done in the early days of looking at bar domains. And so we saw the same thing that everybody else saw, which is if you overexpress this protein sufficiently, you see all these little tubules, this big kind of meshwork of tubules protruding into the cytosol. And it's thought that the reason for that is that you've so overcome the endocytic machinery. You have so much more of the M-bar domain than of any other endocytic protein that you've run out of all the other adapters. And instead of making a bunch of endocytic vesicles, you just see these tubules. But then if you express the full-length protein, you don't see nearly as many tubules or persistent tubules. Instead, you see all these little fragments or maybe short tubules that extend and then get cut off or retract. So that's sort of in line with our thinking, but a key caveat here is that the disorder domain here of amphiphysin can recruit the whole protein scission machinery. So it can recruit dynamin, which is still in these cells. So it's possible that we're just recruiting the proteins that are going to cut the tubules off. So to check that out, we made another chimera where we replaced this disorder domain with a disorder domain that isn't involved in endocytosis. Some of you all might be familiar with the neurofilament domains, which are disorder domains that have been studied for a couple of decades now. And interestingly, these domains play a steric role in cells. So they're sort of these brushes of disordered proteins that extend off of these neurofilaments. You can kind of think of them like a pipe cleaner or like one of those bottle brush flowers. And their job is to space out the neurofilaments and therefore control the caliber or the sort of the waistline of an axon. So we took these domains, which are not in this type of cell at all, and fused them to the I-bar domain, or sorry, to the N-bar domain of amphiphysin. And then, so when we express this chimera here, we saw a similar phenotype to what we get with the full-length protein, which is these short tubules or very, very few tubules, mostly just fragments. Okay, so from these data, we can observe that the presence of the full-length or of the disorder domains bound to amphiphysin's N-bar is inconsistent with the assembly of these long-range scaffolds. So in this way, we kind of changed our thinking about amphiphysin. Rather than thinking of amphiphysin as a protein that stabilizes cylindrical geometries, it now seems more likely that it's part of the fission machinery. It's helping dynamin to cut off these endocytic vesicles, and that function relies on steric pressure among the bar domains, or sorry, among the disorder domains. So in doing this, we were curious, which mechanism is more potent at bending the membrane? Is it the structure-based scaffolding of the membrane or this kind of entropic crowding among the disorder domains? And to address that, Wilton had a clever idea. He said, well, remember, the bar domain family includes these I-bar domains that have the opposite curvature. So we can essentially fuse the I-bar domain to the disorder domains, and we can make a molecule that's really at war with itself. So here's this crazy chimera here, and you can see that part of it would like to bend the membrane downward like this. And then part of it, the crowding among these two disorder domains, would like to bend the membrane outward like this. So we decided to put this thing on the membrane and see which mechanism wins. So first, the control experiments here. So these are giant unilamellar vesicles. They're big spheres of lipid that are just one layer thick. And this is what they look like before you add any protein to them. And if you expose them to the I-bar domain, it'll bind the surface and generate all these inward tubules. And then in contrast, if you expose them to the N-bar domain, the one we were just talking about, it'll bind to the surface and give you all these outward tubules. So then what's going to happen with our chimera? And it turns out to be something of a tie. We see what we call these frustrated fluctuations, where you've got this membrane that's sort of bending outward, and as soon as it bends outward, it'll snap back inward. And that's kind of telling us that these two mechanisms, one structure-based, one coming from disordered proteins and more entropic in character, are sort of on equal footing and can oppose each other. But then when you think about a molecule like amphiphysin, which couples them together toward the same goal, you can understand why that protein is so potent at remodeling the membrane. Okay, so that's what I wanted to tell you about bar domains. But in this next little bit, we'll move on to talking about if disordered proteins are able to bend membranes, then maybe they could also sense membrane curvature. And sensing membrane curvature is really important because it's the mechanism that's responsible for bringing together a critical number of remodeling proteins at the same place, is that they can sense the curvature and sort of selectively assemble together at curved sites. So this is the work of Wade Zeno, a postdoc in the lab who's about to leave us. And Wade did some clever experiments where basically he ran experiments where he tethers vesicles of many different diameters to a cover slip using this chemistry that you can see here. And this technique's been around for a while and has been used to measure curvature sensitivity by structured proteins. And then he just looks at the partitioning of proteins among the small vesicles, medium vesicles, large vesicles. And that turns out to be a really effective means of measuring curvature sensitivity. So when Wade first started working on this, he started by using some structured proteins with well-known curvature sensing abilities. So this again is that structure domain of epsin. And it's been known for at least a decade now that this amphipathic helix is capable of sensing curvature. And if you take this helix and replace it with some tag that doesn't insert into the membrane, so here we're just using a histidine tag, you'll lose that curvature sensitivity. And you can kind of see that in these pictures. So if you look closely, this is the lipid channel, and then here's the protein channel. And then let's take this protein where we don't have curvature sensitivity. When you merge these two together, all the vesicles are sort of a similar shade of yellow. In contrast, for this one that has the helix and senses curvature, you can see that some of the smaller vesicles are a bit more red, and the larger vesicles are a bit more green, meaning that protein to lipid ratio is higher on the smaller vesicles, which is sort of a hint that curvature sensing is going on. So then if we apply one of our disordered domains, so this is the disordered portion of another adapter protein in the clathrin pathway called AP180. And you can see again here, when you merge the two images, you get this heterogeneity in the coloration that tells you that the protein to lipid ratio is not the same on all the vesicles. So all this can be quantified by analyzing the images and applying some statistics. And so here you can see that the control protein here that doesn't have the inserting helix isn't very sensitive to curvature, pretty much the same partitioning across vesicles of all these different diameters. Whereas in contrast, this structured protein as well as the disordered protein are both roughly 8 or 10 times more likely to bind to a very small vesicle with, say, a 20 nanometer diameter than they are to bind to a flatter, bigger vesicle with a 200 nanometer diameter. Okay, but what then is the mechanism of curvature sensing? So to think about that, let's look a minute at this AP180 domain. There's two interesting characteristics physically of this domain. One of them is that it's pretty long, 571 residues, makes it one of the longer disordered proteins in the clathrin pathway. And then it has a mild net negative charge, 0.06 negative charges per residue on average. So not a strong charge, but some charge. So we can think of at least two possible mechanisms. So one mechanism might just come from the long length of this protein. You can imagine that a polymer with that many segments is quite flexible, has a lot of conformational entropy. And when you take it out of solution and ask it to bind to the membrane surface, you're depriving it of many of the orientations that it could have. So there's a substantial entropic cost to this event. But then the more you bend the membrane away from the protein, the more conformations the protein recovers, and so that entropic cost should be reduced. So it's less costly to bind this highly curved small diameter vesicle than to bind this flatter, larger vesicle. Then a second possible mechanism could potentially arise from electrostatic repulsion. Many membranes, including the plasma membrane, the inner plasma membrane of cells, are substantially negatively charged. So if this domain is also negatively charged, you can imagine that there will be a repulsion between the domain and the membrane. And the more the membrane bends away from the protein, that electrostatic repulsion will be reduced. And there should be a preference, therefore, for the more curved membrane. So an interesting way to potentially differentiate between these two mechanisms and figure out which one is responsible is to vary the salt concentration. So if you make the salt concentration very high, you'll screen out the electrostatics. So you would expect this mechanism here to become weaker at high salt concentration. In contrast, making the salt concentration very high makes the polymer floppier because you're screening out the electrostatics. So it increases the entropy of the domain. So doing that ought to make this entropic mechanism stronger. Okay, so down here there's a little table to tell you what do we expect if this mechanism is dominant versus if this mechanism is dominant. So we ran this experiment where we varied the salt concentration between 10 nanomolar and 450, or sorry, 10 millimolar and 450 millimolar, and measured the curvature sensitivity. And indeed, you can see that for this protein, the higher the salt concentration, the higher the curvature sensitivity, which leads us to believe that this protein, its sensing mechanism is primarily entropic. And this was sort of in line with simulation results from Dave Thurumai's lab. Some of you guys may know Dave. He's also here at UT Austin and has been a great collaborator for us. But is this always true? Are disordered proteins always sensing curvature through an entropic mechanism? We thought not necessarily. There should be conditions under which the electrostatic mechanism could dominate. So to try to create those conditions, we took just the first 1/3 of this domain. So that first 1/3 happens to have most of the negatively charged residues. So the charge per residue is about twice as high in this 1/3. And then it's shorter, so it has less entropy, fewer configurations compared to the full-length domain. So here, we did see the opposite. So here, the highest curvature sensitivity is achieved at the lowest salt concentration. So this seems to be a regime in which the electrostatic mechanism dominates. And then, as you might expect, if you look at some intermediate portion, so here's 2/3 of the 2/3 N-terminal fragment of AP180's C-terminal domain, which has an intermediate electrostatic charge per residue, as well as an intermediate length, of course. And here you can see that the curvature sensitivity is almost invariant with salt concentration, probably because of a balance between the electrostatic mechanism and the entropic mechanism. So to kind of summarize that, it seems that long disordered proteins with relatively low net charge will likely interact with surfaces and sense their curvature through an entropic mechanism. Whereas in contrast, if a disordered protein is relatively short and charged, it's likely to interact with membranes through an electrostatic mechanism. And I think this is an unusually large disordered domain with a rather low net charge. So probably this second electrostatic mechanism will be applicable to more proteins outside of this one field. Okay, but you can also see this disorder or this curvature sensing mechanism in cells. So here we're looking at filopodia, these little tubular projections off the edge of the plasma membrane. And we expressed this disorder domain on transmembrane protein chimera. And we compared it to these chimeras that don't have a disordered domain. So here, the red channel is always reserved for the one without the disordered domain. So when neither protein has a disordered domain, you can see the filopodia just look kind of yellow, and the relative partitioning between the plasma membrane and the filopodia is the same in both channels. So there's neither one of these proteins is more curvature sensitive than the other, which is what you'd expect since they're both just fluorescent proteins tagged to the membrane. But in contrast, when you express this disordered domain, you see some positive partitioning to the filopodia, which makes the filopodia look green. And then you can also think about, well, what would happen if you put the disordered domain on the other side of the membrane? Now, we can't do that with these proteins because, with these endocytic proteins, because they would bind all their endocytic binding partners. So we use that same neurofilament chimera, and we put it both outside as well as inside. And when it's outside, it does the same thing as we saw with AP180. It makes the filopodia look green. So it's sensing their curvature and preferentially localizing to them. But if we put it on the inside, instead it wants to move away from the filopodia, and you see them here looking a little red. So it seems that even in the complex environment of the native plasma membrane, these disordered proteins can express a curvature preference. So you might be curious, what will happen if you combine a structured protein and a disordered protein, since that's what most of our natural endocytic adapter proteins do. So again, here's epsin. It's got its disordered domain and its structured domain, and the same thing with amphiphysin. So we measured some of these interactions, and interestingly, what we found is some pretty strong synergistic effects. So here, for example, with epsin, you can see how much curvature sensitivity the structured domain has here in this red curve. And then the green curve is showing you the disordered domain alone. But then when you put them together, you're on this purple curve, which gives you more curvature sensitivity than you would get from just adding these together. And then it's even stronger over here with amphiphysin, that neither one of its component parts is all that sensitive, but the full-length protein is highly sensitive. And we don't fully understand that, but sort of our hypothesis is that this dimer brings these two disordered domains so close together that now not only do they have to avoid the surface, they have to avoid each other. And so there's a really strong preference for binding a really curved vesicle that will provide more volume above the membrane surface for these disordered domains to get away from the surface and get away from each other. Okay, so that brings me to the second part where I'm going to talk about endocytic protein networks. Maybe I'll stop for just a minute and see if anybody has any thoughts or questions before we move into the second bit.
Is there any role of ATP or GTP in this part?
That's a good question. So endocytosis is a pathway in which all the hydrolysis of ATP and GTP occurs at the end. So the machinery responsible for uncoating the vesicle consumes energy in that way. So you have these chaperone proteins that go in and take apart clathrin, and they also convert so most of these proteins that I've been talking about bind the lipid PI45P2. So energy comes into the pathway directly by two means. One of them is uncoating, and the other is converting the lipids into phosphoinososides that the proteins no longer have affinity for. So all those costs are paid at the end to take the vesicle apart. But the process of assembling the vesicle is all spontaneous or all energetically downhill. There's no ATP hydrolysis involved in vesiculation. Does that answer your question?
Yeah, thank you.
Sure. Okay. So yeah, sure, go ahead.
Yeah, I'm just curious if you ever try to swap instead of the disordered domain by another disordered domain, the disordered domain with an ordered domain, and see if you have the same phenotype where you have like smaller vesicles, or if it's something that is a property of the disordered domains.
Yeah, that's a good question. So for the first bit about bending, before we ever got interested in disordered proteins, we had studied how these steric or crowding effects can drive vesiculation using always structured domains. And then at some point we realized that the largest volume occupancy or the sort of the largest domains in terms of how much space they take up and how much steric pressure they could generate in this pathway are the disordered domains. And that was actually what got our lab interested in the disordered proteins, is their large hydrodynamic radii. But you can certainly see those same effects with structured proteins if you can find structured proteins that are large enough. Many of these disordered proteins, for example, amphiphysin's C-terminal domain has a hydrodynamic radius of almost 10 nanometers. So it's rare to find a structured protein that's that large. But I think the effects would be the same if you could. They might even be a bit stronger because the disordered protein is flexible and can change its shape, of course, even if that's entropically disfavored, whereas a structured domain is, of course, more rigid. Then in terms of curvature sensing, there I think the disordered domain really does bring something unique. So of course, you can just imagine that if you have a vesicle that's small versus one that's large, and you attach a structured protein of a certain diameter, that the smaller the vesicle is, the more proteins you can pack around it, right? It's like runners on a track. If you happen to get the inside lane, you have not as far to go, right? So there's more and more space the further out you go. So you should have some curvature sensitivity by very large structured proteins. And we can measure that in our experiments, but it should be very predictable and quite linear with with decreasing radius. Whereas the disordered domains all seem to sense the curvature of the membrane in a way that's more significant than that. So I think there, these electrostatic and entropic effects are really enhancing curvature sensing.
Thank you.
Great, good questions. Okay, well, if you think of more, just let me know. But we'll move into this second bit here, which gets us into condensates, which is the area where you guys probably have the most direct interest. Okay, so I should tell you, if you're not, if you don't regularly think about endocytosis and membrane traffic, that it's an incredibly complex pathway. There's at least 30, possibly as many as 50 proteins involved in forming a clathrin-coated vesicle. And almost all of those, N-1, are adapter proteins, and clathrin coat being sort of the same regardless of the mixture of adapter proteins. So it's long been a question, why do we have so many adapter proteins? What are all these adapter proteins doing? And it's really tough to think about reconstituting this whole complex pathway with that many proteins involved in it. But at the same time, if by just studying one protein at a time, we're obviously missing very important, possibly dominant interactions. And so we really wanted to do more in terms of trying to study protein-protein interactions, but we were kind of in a quandary about how can we do that meaningfully given that there's so many proteins in the pathway. So what we decided to do was look at some of the very earliest proteins. So very early in the pathway, there's relatively few players involved, so we thought that'll be the easiest way to start thinking about protein-protein interactions. And fortunately, since this pathway has been studied for so many years, it's really been nicely mapped where we know which are the first proteins to arrive, then what's sort of the second wave and the third wave, and so on and so forth. So in this pathway, the first wave, which has been characterized in all these references you can see on the slide, is the arrival of what we call this pioneer complex or the initiator proteins. And this complex consists of a couple of proteins. One is EPS15, and the other is FCHO1, or an FCHO1 has a second isoform, FCHO2, and EPS15 also has another isoform called EPS15R. But these two families, EPS15 and FCHO. So both of these proteins are dimers. FCHO, we've already talked about the F bar, so that's that more shallow bar domain. And it has a morphology that looks a lot like amphiphysin. It's got this disordered middle domain, and then its C-terminal domain, rather than being an SH3 domain, like it was in amphiphysin, is this thing called a mu-HD domain. And that mu-HD domain interacts with the disordered portion of EPS15 in this complex that you can see over here. So then EPS15, we haven't introduced yet, it has these EH domains, and they are PIP binders, as is the F bar. So these domains all stick to the membrane by interacting with PI45P2. But then EPS15 has this coiled coil domain, and then it has this disorder domain that interacts with FCHO, and the disorder domain also can bind back to the EH domains. So this protein has multiple ways of generating a multivalent network. Okay, so we started looking at these proteins and trying to figure out what they would do if they were together on the membrane surface. And this is the work of two people in the lab, Casey Day, who's a postdoc, and Grace Coggo, a graduate student. So Casey and Grace, they started doing this work, and at first they made these giant vesicles, and they purified full-length FCHO and EPS15, which at least from mammalian cells had not been done before. So that was a feat in itself. And we have a lot of help on that from our collaborator, Eileen Lafer. So these proteins by themselves are not very interesting. We add them to giant unilamellar vesicles. You can see a cross section through the vesicle as well as a 3D projection. And there's not really any interesting arrangement going on here, really just a uniform coating of the membrane. But then if you add them at the same time to the membrane, you get this interesting partitioning between a denser phase here and a more dilute phase. And you can see that in the projection, you see kind of a little cap, like a baseball hat on the vesicle there. And And sometimes rather than seeing just one cap, you'll see a bunch of little spots that wiggle around. But no matter what, you see this partitioning between sort of a brighter phase and a less bright phase. And if you reduce any of the multivalent interactions between these, so if you mutate this disorder domain to make it not bind the mu-HD domain, or if you remove the ability of EPS15 to dimerize, or make various mutations that reduce the affinity here, then you'll not see these domains form nearly as often. So it seems that they depend upon these multivalent interactions. So then giant vesicles are nice because you can see these large domains, but they make it harder to really monitor what's going on in the membrane surface. So we also do these experiments on flat membranes supported by layers. And again, on supported by layers, no assemblies without adding both proteins. But when you do add both proteins, you see these assemblies. And when the assemblies bump into each other, they'll fuse together, indicating some sort of a dynamic behavior. You can also frap them and they'll recover. So this made us think that, you know, there's a lot of the properties we think of in terms of condensates. So we've got intrinsically disordered proteins, dependency on a multivalent network, there's some merging going on and some molecular exchange. So we started to wonder whether these two proteins might be forming some sort of condensate. And I'll say on our part, this was a reluctant conclusion. We had, as you've seen, studied disordered domains independently for years, and we really think about them as sterically repelling each other. So the idea of thinking of them making this condensate was a little foreign to us. But you can see that when we take these proteins in the absence of the membrane and mix them together with a bit of PEG in solution, you do see these droplets kind of raining down onto the cover slip. So here's closer up of the droplets fusing together. And you can frap the droplets and see them recover. So that they seem to have the properties of a lot of the other droplets that you see in the literature. And then once you have this droplet system, you can think about mapping phase diagrams. So you can heat the droplets up and they'll dissolve and go away. And then when you cool down the same solution, you'll see the droplets come back. And we, like many others, have recognized that you can use these images of droplets at different temperatures to make sort of a relative phase diagram. So you can measure the approximate protein concentration inside compared to the approximate protein concentration outside. And that will let you map this sort of arbitrary units phase diagram here. So remember I told you that EPS15 has an ability to make a multivalent network all on its own. And indeed, if you cool EPS15 sufficiently, so here we're below physiological temperature, you can get EPS15 to form droplets. But then if you add a bit of FCHO, the critical temperature will go up substantially. And the more FCHO you add, the more the critical temperature will go up. So it seems that these two proteins are sort of are stabilizing each other in a mutual way, which is sort of in line with what we saw on the membrane surface, that we couldn't see protein aggregation or protein condensates on the membrane surface without having both proteins. So it was really important to us to test what are these structures doing in the cell, if anything. And we had to think pretty hard about how to do that. But Casey had a clever idea. So she said, remember back from that slide where we were doing all the mutations on EPS15, that this coiled-coil domain that makes this protein a dimer is really essential. If we don't have that coiled-coil domain, no matter how cold we go, we never see this EPS15 form any condensates. So she thought, let's borrow this clever idea from this paper by Cliff Brangwyn. I'm sure many of you have seen this, where you can use this Cry2 domain to make a light activated clustering of IDRs and see droplets show up in the cell. So Casey adopted that, and she replaced this coiled-coil domain with Cry2. And so this is still in vitro, but she took this purified construct, and when she shines light on it, she can get these condensates to appear in the presence of the blue light. So then she decided to take this tool into her mammalian cells. So here I have to give you just a moment or two of background on how we look at the dynamics of endocytic events, since a lot of you probably aren't in this field of studying endocytosis. So here is the plasma membrane under turf illumination of a mammalian cell. And this is a sum cell, another cell with this really broadly spread, another epithelial cell. And it's also one that's pretty easy to gene edit, which is the reason we chose it for this experiment. So here, this cell line originally came from Tommy Kirkhausen, so we thank him for it. And he had used gene editing techniques to put a halo tag on AP2. And AP2 is one of the major adapters of the clathrin-coated pit, so it's sort of a clathrin-coated pit marker. And so you can see all these coded pits here, and then we've labeled we've labeled EPS15 with M-cherry. So if you fix your eyes on any one of these little dots, you'll see it show up and get brighter and then eventually go away. And here we've zoomed in on just one, and you can see it go through that cycle of appearing, getting brighter, and then going away. So these are these endocytic events, and at the plasma membrane, the average endocytic event lasts about a minute, but they can vary from 20 seconds to 2 or 3 minutes. And endocytic pits can basically be categorized into one of 3 outcomes. So some pits are there at the plasma membrane so briefly, usually under 20 seconds, that it's been shown that they don't actually capture any cargo and they never form a vesicle. They're just stochastic assemblies of adapters and clathrin that then disassemble. And then at the other extreme, clathrin-coated structures that stay at the membrane for more than about 3 minutes. If you follow them, you'll find out they'll stay there for tens of minutes. And these are what we call stalled structures. So they're not really productive. They're just staying at the plasma membrane. And there have been some recent work suggesting that these structures have roles in signaling, maybe doing things other than endocytosis. But then the majority of the structures are in the middle, and they'll form a productive vesicle and depart anywhere from 30 seconds to a couple of minutes. So in wild type cells, this is a typical distribution. So a small fraction of coated pits are in this abortive category, a few are in the stalled, and most of them are productive. So this is in cells expressing the wild type version of EPS15. But then when you knock out EPS15, the main change that you have is that there's an increase in these abortive structures. And that kind of makes sense because EPS15 is thought of as one of the main catalytic proteins for getting endocytosis started. So if one of those catalysts is missing, you're going to have more structures that don't get off to a successful start and ultimately abort. So then Casey expressed this Cry2 chimera here, but she was careful not to expose the protein to any blue light. And here you'll see that abortive phenotype is maintained. In fact, it's maybe even increased a little bit, possibly because this protein is blocking sites that could be occupied by compensatory proteins. But then when you shine some blue light on these cells, you can recover a fairly close to the wild type distribution of dynamics. So you can see the abortives go back down close to wild type levels. So at this point we thought, oh, this is exciting. We'll keep shining this blue light and we'll drive the abortives down to 0 and make endocytosis more efficient. We thought this would be really fun. But interestingly, when we turned up the light, we didn't quite get what we expected. We did get a small reduction in the number of abortive pits, but most notably, we got this huge increase, about a 4-fold increase in the number of stalled structures. And you can see the stalled structures in this video here. You see these large endocytic structures that some of them, if you fix your eyes to them, you'll see they persist through the whole movie. They don't, and this is, I think, a 10-minute movie. So they're clearly lasting a lot longer than a normal endocytic structure. So what's going on with that? Why is more not better? We were really perplexed by this, so we went back to our droplets and we put different levels of blue light on them. And I'll let this movie cycle around again. What we saw was that the level of blue light that had been giving us a rescue of dynamics gave very fluid droplets, whereas this level of blue light that caused the stalled structures was giving us still nice rounded droplets, but they didn't fuse. They formed these kind of gel-like structures where you can see 2 that have stuck together, never re-round over a long period of time of multiple minutes. Okay, so we also saw that if we frap these same droplets, the recovery time of the droplets formed under low light was very similar to what we get with wild type EPS15. Whereas in contrast, when we apply that strong light, we got a much slower frap recovery time. So we began to think of this problem kind of like a classic catalyst working in a reaction. So EPS15 and FCHO are these early catalytic proteins. And like all catalysts, they really have 2 jobs they have to accomplish. They need to bring together reactants And we've been kind of focusing on that function, thinking more blue light will be better. But they also have to release the product. And in this case, the product is the coated vesicle. So if the coated vesicle can't be released, then the catalyst is not efficient. So we began to wonder whether this fluid light catalytic complex really gives us a balance between these two functions, that we're still efficient in bringing together reactants, but we're allowing the rearrangements that are necessary for release of the vesicle. So to kind of think a little bit deeper about this, we looked at the next adapter that's recruited after EPS15 and FCHO assemble, and that is AP2, which I depicted here in this little yellow cartoon. So here we frapped AP2 in the presence of these different light levels that had driven assembly of EPS15 Cry2. And what you can see is that the wild type recovery of AP2, so AP2 in the presence of wild type EPS15 is this black curve. And if we put in Cry2 EPS15 and don't apply any light, we get too rapid recovery of AP2, as if AP2 isn't being drawn to the pit long enough to drive a productive membrane vesiculation event. But in contrast, the low light level brought us back to sort of the wild type level of exchange of AP2. But if we keep going and make EPS15 2 and FCHO 2 solidly assembled, then AP2 spends too much time at the coated vesicle, indicating that it probably can't rearrange enough to drive the membrane remodeling and the formation of the vesicle. Okay. All right, so that was a lot. I hope I didn't overwhelm you with too much information, but just to conclude here, I'll say we've talked about how disordered proteins can drive and sense membrane curvature, and how coupling them to an ordered protein like a bar domain can really lead to some synergy where the structured portion and the disordered portion are working together to bend and remodel the membrane. And then that these protein droplets, we're really still learning about what their function is in endocytosis, but they may be providing a fluid and therefore really efficient and flexible catalytic platform for initiating endocytosis. And I'll stop there and thank my students who really have made all of this happen. So the first bit was for Wilton Sneed, who's not in this picture. He recently moved on, but then we talked about the work of Wade Zeno and primarily Casey Day. All right, thanks guys, and please let me hear your questions. Well, I don't know, I'll see if we should stop sharing or not. Maybe we can for a minute and see if you want to go back to any of the slides.
Thank you so much. That was wonderful.
Well, thank you.
Yeah, beautiful stuff. Really wonderful. I keep thinking about the energy, the binding energy that drives the formation of these dimers.
Yeah.
You know, I wonder, because each monomer is already embedded in a membrane, so entropically they're near each other to begin with. So I wonder if they're fairly weak.
So they seem to be strong. We can take
strong.
We can take either protein and dilute it. If you're familiar with FCS, you know, you run those experiments under very dilute conditions, you know, 10 nanomolar. And we'll still measure a big hydrodynamic radius compared to if we truncate that domain and measure the monomer. And everybody has always said that, well, you can kind of see it in the structure of the bar domain. You've got these long alpha helices that are really too long. Like a bar domain as a monomer, those helices would bend, but or would kink. But because they overlap each other, they're stable. So you have such an enormous overlap. I think that's what makes the KD so low for the dimer.
Yeah, it's really nice. It's so fundamental, and the way you study these systems is so beautiful. And these are such complex systems, and you've dissected them so beautifully.
Oh, thank you. I appreciate that.
I have a question about cargo. So
Sure.
When the cell need to form some kind of vesicle, I'm thinking the purpose must be transporting some kind of cargo, right?
Yes.
Protein, mainly protein. Maybe also use that to transport some lipid, but it's mainly protein. So how does the cargo play a role in forming the clustering coated pits? How does it know, okay, I have a vesicle that is full of angiotensin receptor or full of protopump. So how does cargo,
That's a great question. Yeah, I'll tell you what I know about it. So so these cargo adapters, most of them, they bind three things: lipids, a cargo, which is a transmembrane protein in the terminology of this field, and clathrin. And we didn't talk about cargo, as you pointed out. So most cargo have a tail that extends into the cytoplasm, and that can be anything from a very short motif to a large folded domain. But specific biochemical motifs, they're usually 4-letter codes. Like the most classic one is YXX5. So you've got 2 amino acids that don't matter, and then the phi refers to something hydrophobic, and then the Y on the other end. So that motif will recognize AP2, which is the best studied adapter in the clathrin pathway. And there have been studies that have shown that if you overexpress a cargo, that will increase the recruitment of AP2 to the membrane and give you more endocytic events per area of the plasma membrane. So in that sense, endocytosis is cargo dependent. But the increase you'll get is not particularly large, maybe 20, 30%. You're not going to double or increase the number of endocytic events by tenfold. Endocytosis is largely constitutive. It'll go on and on, even if the number of cargo per vesicle is relatively low, or if the number of cargo is relatively high. And I think that is less understood exactly. How is it that endocytic vesicles are as cargo independent as they are? Is an ongoing area. We had a project for a while where we were trying really hard to see what it would take to stop a coated vesicle. So we made these cargo with these ridiculous disordered domains displayed on the outer surface of the cell. And we would find vesicles where they were very crowded together, and we were trying to generate some steric pressure to oppose endocytosis. And it turns out that's very hard to do. Mostly what you get under those circumstances is just fewer cargo per vesicle. It seems that this sort of interwoven network of adapters on the coat side, there's such a high energetic barrier to disassembling that almost nothing a cargo can do, whether it clusters or is very large, will really even slow down a coated vesicle, which is kind of curious. We're still kind of trying to understand how that pathway is as robust as it is.
Thank you. That's fascinating.
Thank you.
Yeah,
Oh, go ahead, Avi.
Oh, sorry, Caroline, you go ahead. You were first.
No, you go first. You got your video on.
Okay. So I found the phase diagram quite striking, right? So if you look at the shape, it looks like, not kind of like a binodal as you would expect, right? One side looks like more close to a kind of spinodal straight, and one is more gradual, more like a binodal. Any comments on what might be determining such a,
You guys are the phase diagram experts. Maybe I'll pull it back up again here, and you can tell me what it means to you, because we have noticed that and not really known how to interpret it. But I'll bring it back so you can get a proper stare at it. Let's see, to figure out how to efficiently advance my slides. It's not too far back here. Oh wait, there it was, sorry. Got carried away. Okay, yeah, there you have it. So you're commenting on this side being very straight, and whereas this one is much more tapered.
Exactly. So yeah, I don't know how to interpret that, right? It seems that you somehow, and this is in the presence of, are these just binary components, both the FCHO1 and EPS?
That's right. Or no, they are both dimers.
Yeah.
Yes, but you're right. This is binary. So there's only 2 proteins here, and this isn't the Cry2 system.
Yeah.
I guess this is over here is telling us that the droplet itself has just sort of one concentration. Its concentration doesn't change very much as we increase temperature.
Exactly. So
whereas the concentration of the liquid's varying quite a lot.
Yeah, that's pretty pretty interesting. That is something at least I haven't seen before.
And if you know what to make of it, tell me. I know an alternative, you know, when I see something interesting, I first ask, can it be an artifact? And, you know, here we really just wanted to know the shift in the critical temperature, but the way we're measuring this is just by comparing relative brightnesses. So I guess I don't immediately see how this can lead us astray, but it's worth thinking about. You know, would we expect our camera to be, though I think all these images are pretty, none of them are anywhere close to saturating the camera, and we have a nice linearized EMCCD. So it's not immediately apparent to me how we're measuring it wrong, though, you know, I want to think more about that. Should we really trust how straight this is?
Yeah, maybe one other way would be maybe complementing it with the alternative non-fluorescence based measurement, right?
Yeah, and we have trouble getting enough of this protein to do a more bulk measurement, but we could do FCS. So, you know, all experiments are tricky. That one will be a little tricky to get it just dilute enough in this phase, but that can be accomplished with enough fiddling around. You know, another possibility maybe we need to, well, no, I was about to say maybe we need to plot this differently, that there is a slope to these, but it's just so overwhelmed by this slope. But that in itself is what's odd here.
Yeah.
Yeah. If you have any theory about what that could mean, let me know.
Yeah, sure.
I'm glad to learn from you.
Yeah. Okay.
I'm going to go out of sharing.
Thank you so much for this talk. It was really interesting. I wanted to ask a general question. In the first story, you were talking about the lipid that all the proteins are interacting with. Can you tell us a little bit about how you identified that lipid, or how you confirm? Was it P154?
P145, P2? Oh, okay, yeah. So it's not me who identified it. So this is quite an old field. So people have been studying these adapters for at least 20 years, maybe 30. And the early experiments that were done, people would take different phosphonosticides in that pathway, PI4P, PI45P2, PI345P3, all the ones that make up this pathway of the metabolism of PIPs. And they would basically do like Western blots against PIPs. Okay, you can still buy these things today, the so-called PIP strips. Avanti will sell them to you. And also, it's known that the predominant PIP at the plasma membrane is PI45P2. So it also kind of makes sense that these proteins would bind PI45P2. And then, you know, as the vesicle progresses, so we think of every organelle, whether it's the plasma membrane or the endosome or the lysosome, as having its own like PIP signature. So gradually the PI45P2 is turned into PI4P to make this thing look like an endosome, and then endosomes don't bind these proteins that are plasma membrane proteins, which is all the adapters. So the adapters will hop off, and then these chaperones take apart clathrin, and that's how you uncoat the vesicle. So yeah, PI45P2 is been studied by a lot of folks and is sort of the plasma membrane PIP.
Great, thank you.
Sure.
So, hi. I had a sort of a technical question.
Sure.
So in your Cry2 experiments, the in vitro ones where you over illuminated the drops and they fell into the gel-like state.
Yeah.
Once you remove the light, do they dissolve, or they stay as a gel?
The ones that are more fluid, if you remove the light, they'll disassemble.
Right.
But the ones that are more gel-like, when you remove the light, they don't disassemble nearly as much. They might get a little smaller or dimmer, but you won't really see them totally go away.
That's interesting. So could the stalled sort of be acting as a kind of a sink, just sucking out all those proteins that are
That's a good
in
thought.
the cell experiments?
Yeah, that's true. So we run the experiment, we have sort of a fixed duty cycle of the light, right? And meaning how often we're flashing and for how long. And that is one question we've had. Like, our movies are relatively short, so I'm not sure whether we're reaching an equilibrium. Like with the more fluid one, the lower light levels, you might think, okay, if you go on long enough, you'll probably reach some equilibrium level of assembly. But if you have a protein that's forming a gel-like structure, then you're sucking all the protein to the membrane might be your equilibrium. And that that's probably part of this this stall, is not just the slow recovery, but also just the growth of the thing. And you can see some large endocytic structures by the end of that movie. So yeah, I think that probably does play a role. One experiment we've wanted to do is to, flash the light for half the movie and then turn it off and see what happens. Like, I bet we would find that with the low light level, we'd go back to our underlying dynamics that we had before, which would be the dynamics of the knockout. Whereas with the high light level, we at least wouldn't relax that far, if at all. That makes me more motivated to try that experiment.
Thank you.
Sure, thank you.
So I have one other thought, just really listening to the whole talk, is I'm certainly not remotely an expert on this, but I've heard other people who've thought deeply about the physics of a tumor talk about the pressure that tumors are under. So I was reading some work, for example, from a professor named Liam Holt, who's up in New York at NYU, who studied, for example, how in pancreatic cancer, the actual tumor cells are under tremendous pressure. And so I'm just thinking out loud here, I'm wondering whether the mechanics of membrane maintenance are somehow different.
That's an interesting thought. So one field in which that is really intersects with this is yeast. So yeast, as you know, have this turgor pressure. They would explode if they could, if it weren't for their big coat. Yeah. And it's been, so endocytosis was probably first studied in yeast because of the ease of genetic manipulation in yeast. And there's still many people who study endocytosis in yeast. And what they have found is that to overcome that turgor pressure, actin becomes essential to endocytosis. So the bud starts to form, and then actin assembly is what pushes the bud in and allows it to actually complete. And in yeast, if you inhibit actin, endocytosis is dramatically knocked down. Whereas in mammalian cells, you can inhibit actin, and at least in the short term, endocytosis barely notices. Though, further, it's been observed that if you stretch a mammalian cell, so people have actually plated cells on these like flexible substrates and stretched, then you can induce an actin dependence, even in a mammalian cell. So that's interesting to think about that. Yeah, it'd be probably pretty simple to see. Has anybody ever measured actin dependence in tumor cells of endocytosis? Of course, all of us are studying tumor cells, whether we like it or not, unless we're trying really hard not to. But I don't think our cells in that have that same pressure, right? That's not just being a tumor cell, it's the physiology of the tumor.
Yes, yes.
And with that pressure, I've heard of like tumors are under pressure, and if you try to, like a part of my work is in drug delivery, which I didn't talk about at all, but when we're thinking about that, we worry about, the particles we're creating are going to be flushed out of the tumor. So fluids flowing out of the tumor, right? So I have to think about the geometry of that. What's that doing to the membrane? Does it stretch or compress?
Yeah, it's some interesting tensegrity balance. I don't know exactly how it works, but yeah, it's funny because
imagine, maybe the tumor, if there's fluid that's trying to get in there, the whole thing is sort of stretched. Yeah, so membrane tension probably is higher in tumors. I wonder how much is known about that.
Yeah, I think it probably is a whole separate field. It's amazing how, everywhere you look in biology, things are more complicated than you think. I mean, a membrane seems like it's such a simple thing, but of course,
Oh yeah, there's this
that's
guy
nonsense.
Where is he? I think University of Iowa. He's got a Turkish name, Komert Kurl. I always mix up his first and last, but he was out of Tommy Kirkhausen's lab, and he studied, has been studying endocytic pit dynamics during development. So in that context, he sees cells that are like undergoing morphogenesis. So the membranes are stretched while these cells are moving, and he's measuring endocytosis like during that. And he has found some interesting effects there.
Yeah, it's beautiful. I'll look him up.
Yeah, I think he's Iowa. Is that right?
Iowa, okay.
No, Ohio.
Ohio, okay.
The Midwesterners will be annoyed with me to mix up Iowa and Ohio.
Yeah. So, other questions? Deanna, anything else?
I don't think so. Yeah, no, I think I'm out of questions.
Well, thank you guys so much.
Oh, it's been
Yeah, thank you.
wonderful. Thank you for doing this. It's been a
it's
load of fun, and we may have other questions that we'll follow up with you, but
that'd be great.
Now,
yeah.
For now, I think we're good. All right,
Okay,
thanks
great.
again.
Great.
Yeah,
Have a
thank
good
you so
one.
much.
Okay, you
Bye.
take care. Okay, thank you.
Bye.
Bye bye.